Today we'll talk about viruses, their classification, and lab diagnosis. Let's start. So, viruses can be classified in two ways. First one is classical taxonomy, and the second one is by their genome and the way in which they create mRNA. mRNA is created in the host cell by ribosomes of the host cell. So the classical taxonomy will start not as in regular taxonomy of other organisms and microorganisms. So we will not start it from domain because we cannot really call the viruses as living organisms. So we'll start with order. The order will have in its name determination viralis, the family with termination viride, subfamilies virine, genus, virus, and species and serotypes. But what we need to notice that not all of families will have subfamily and not all species will have serotype. Talking about Baltimore taxonomy or Baltimore method of classification of viruses, we have 7 classes or 7 groups. So group 1 will be double-stranded DNA virus. Group 2 will be single-stranded DNA virus. Group 3 will be double-stranded RNA virus. Group 4 will be positive sense single-stranded RNA virus. Group 5 will be negative sense single-stranded RNA virus. Group 6 will be the single-stranded RNA virus. And group 7 will be double-stranded. Group 6 will be the single-stranded RNA virus that are retrotranscribing. And group 7 will be the double-stranded DNA virus that are viruses that are retrotranscribing. So these are so-called retroviruses. Regarding the size and the shape, as well as every kind of microorganism, if we can call the virus microorganism, they are composed of different chemical structures, and they have different size and they have different shape. So we can imagine that the biggest virus can be 100 times smaller than bacteria and 1,000 times smaller than eukaryotic cell, for example, human body cell. And the smallest viruses will be even smaller, like not just thousands but millions times smaller than bacteria and eukaryotic cells. Also, viruses will have different shape, and the shape will be determined by different structures. For example, we can have naked viruses and enveloped viruses. Naked viruses are covered just with capsid, and enveloped virus is covered with envelope or supercapsid. So what determines the shape of the virus, it is actually the chemical structure of capsomeres, and capsomer is a kind of proteic substance, proteic structure, which makes these particles. It's a unit which unites other parts of the viral capsid. And also on its surface, viruses can have specific receptors which will have their antigenic structure. For example, here you can see coronavirus, which is today making a worldwide problem, which is called pandemic. Well, right now we have the pandemic caused by this virus. So independence of the shape and of the structure of the capsomer, all the viruses will have different shape and different size. Talking about cultivation of viruses, we can describe three methods. The first method is in vitro cultivation. In vitro is translated inside the glass. And actually in the medicine, in vitro is called every reaction which happens outside the living organism, but inside the glass, because usually it happens or in the tube or in the Petri plate or in special boxes. So this one is the special box for tissue culture. Well, for such purposes, we are using specific tissue cultures, and to make it, as well as we know that all the viruses, they have specific affinity to specific cells. As a tissue culture, it is better to use stem cells, which are universal cells, which are mother cells for every kind of human cells. For example, it can be the product of abortion. It can be used for this purpose. Why we cannot cultivate viruses as bacteria? Because viruses are mandatory intracellular parasites. Well, because viruses are not multiplying by themselves, they have no organella, they are not living microorganisms, they are just intracellular parasites. So even inside the cell, they are not able to multiply by themselves. And here is an example which I like so much, is the question why computer viruses are called viruses but not bacteria. So what is in common between computer viruses and the real viruses in microbiological world? Well, as well as every computer is composed of two parts, the two main components. The first one is hardware and the second one is software. Well, the hardware is everything we can touch in the computer. They are real physical components of the computer. Meanwhile, the software is everything what we cannot touch. So it is operating system. It is about all the programs and all the data we have inside the computer. So all the software is composed on binary codes. As well as we know, these codes are big amount of 0 and 1s. So it is a kind of code which makes the data inside the computer, which makes software inside the computer to be useful. In the same way, every living cell has its own genetical material, which is also composed of codes. So whenever a computer virus is getting inside the computer, it is not able by itself to destroy the computer. But the only thing the computer virus is able to do is to destroy, to change the code of the computer software. So it will change as well the purpose of the computer. If till now the computer was doing everything that the user wanted to do with computer, from this moment the virus will control the action of some software in this computer. The same thing happens in the cell. Whenever the virus is getting inside the cell, it infects the cell and it starts to integrate its own genetical material in the cellular genetical material. It actually will change the purpose of the cell. So one of the main purposes of the cell is to multiply, to produce other cells. But from the moment the virus will infect the cell will start thinking that its purpose is producing viruses. So actually the cell will become the factory of viruses. This is why the virus is not able to reproduce by itself, and this is why the virus will need living cells in order to multiply. So in cultivation methods, first of all, we can use in vitro cultivation. That means it is not in a living organism, but it is in living cells, in living tissues. So in this special box, we will create required conditions for the cell culture, required pH, required temperature, all the required conditions in order to keep this tissue alive outside of the body, and we will be able to inoculate this tissue with viruses. In this way, we will see the multiplication of viruses in the places where the tissue culture will start becoming transparent. That means the cells will start destroying because of reproduction of virus. Because virus is reproducing inside the cell, it breaks out the cell when there is no enough space inside the cell. And in this way, it goes in other cells and contaminates them faster and faster, because in the beginning, just one virus can get inside one cell, but when the cell will explode and will release viruses, it can release hundreds or even thousands of viruses. Well, the second cultivation method is in ovo, which means inoculation of embryonated egg. So here you can see that we can inoculate different parts of the chicken embryo, and it will depend on which kind of viruses we are studying now. And the last one is in vivo cultivation. In vivo means in lab animals. So if the first one is called in vitro, which means outside the living cell, and the last one is called in vivo, which means inside the living organism. Outside the living organism and outside the living organism. But the second one is something intermediary, because embryo is not considered to be a living body yet, but also it is not just a simple tissue. So it is something between. Methods of diagnosis. Every diagnostic method can be divided in direct methods and indirect methods. Well, direct method will be called that method in which we can see the virus itself. So when we can see the virus itself, that means it is direct identification, direct method of diagnosis of viruses. For such purposes, we are using such device as electron microscope. Please don't misuse with the word electric. Don't call electron microscope electric, because it is not about electricity, but it is about the principle of action of electron microscope, and namely, this device is able to send the jet of electrons towards the smear, towards the studied material. And these extremely small particles, which are called electrons, are reflecting from these extremely small viruses, and they are going back to the matrix, which is able to process this extremely small unit, this extremely small virus, and the image is processed by computer and is shown on the screen. So whenever you will see the electron microscopy picture, its initial look is this one. It will be all the time white and black picture. So if you will ever see the colored picture of viruses, you need to understand that this picture were processed after it was taken just to make possible to make difference between different structures, to study different structures of the virus. So regularly the picture of the electron microscope will be looking like this one. And here we can see the picture of electron microscopy of the coronavirus. Another method of diagnosis is serological method. And serology includes a lot of different tests, a lot of different procedures. Some examples of them are neutralization reaction, are precipitation reaction, immunofluorescence assay, immunoenzymatic assay, radioimmune assay, ELISA Western blot, and so on. So for example, here we have a small element of ELISA test. So here we can see the antigen, and as antigen we should understand every foreign protein for our body. So every protein which was not produced inside the body is considered to be antigen. So every foreign protein inside the body, is it bacteria or fungi or virus, every proteic structure will be called antigen. So the viruses will have their own antigenic structure inside and outside of the virus. But antibodies are specific proteins produced by our immune system as response on the attack of antigens. So the purpose of antibodies is to catch and to bind and to destroy antigens. This is a specific kind of immunity. How can we make difference between two ways of serological diagnosis? First one is seroidentification and second one is serodiagnosis. In seroidentification, we are taking known antibodies and unknown antigen. That means we are taking pure culture of the pathogen from the patient, pure culture of the pathogen, whatever it is, virus or bacteria, we are taking it from affected area. So in this case, we can suppose which is the pathogen, but we don't really know which is the pathogen. So in seroidentification, we are taking antigen from the patient, which is pathogen. Pathogen has its antigenic structure, and we take known antibodies within serum. We are taking known antibodies, which we suppose would match with this unknown antigen, and we are making reaction. If reaction will happen, that means seroidentification is positive. By seroidentification, we will understand if this pathogen we took from the patient is that one we supposed or not. In serodiagnosis, we have unknown antibodies and known antigen. So whenever we need to check if the antibodies are present in the blood, we'll take some blood or some serum from the patient. We will take some known antigen, which we suppose for these antibodies, and we'll make reaction between this blood test and this antigen sample. So if the reaction will happen, if the reaction will be positive, we will prove that blood contains antibodies, that immune response happened, and we have antibodies for this very antigen. This is how happens serological test in infectious diagnosis, including viral serological test. And the last method of diagnosis is identification of the viral genome by molecular biology techniques. Namely, it's about PCR, polymerase chain reaction, or the mostly fast technique, real-time polymerase chain reaction. So this test, again, will come back to the topic of coronavirus because it is very important for us right now. Right now we are using real-time polymerase chain reaction to diagnose as fast as possible the infection with this coronavirus. So if we'll describe very briefly the PCR, this test, we can say that in this test, by using specific components, molecular components, we can start multiplication of the genome of this virus. So if and this reaction happens in more cycles. As more cycles will do, as more chains of DNA of this virus will get in the end. So finally, after 30 cycles, we'll have the enough amount of copies of this DNA in order to be able to prove that we have this viral infection right now. So here is the general explication of the methods of diagnosis of viral infections.